When we hear the word cholesterol, our minds usually jump straight to good, bad, and what our doctor tells us about our blood tests. But what if I told you the most incredible cholesterol story is one you've probably never heard? It's a tale happening right now, deep inside every single one of your cells. So let's start with a big question. What if the key to understanding cholesterol's role in our health isn't about what's floating around in our bloodstream, but about what's built into the very walls of our cells? That's where we're going today. Okay, so we're talking about something called membrane cholesterol. This stuff is a superstar. It's not just some passive fat. It's a critical piece of architecture embedded in the outer layer of all your cells, giving them shape and, maybe most importantly, controlling how they talk to each other. And here's the kicker. We've known for a long time that when this cellular cholesterol balance gets messed up, it's connected to huge problems like atherosclerosis, the hardening of the arteries. But the how, the nitty-gritty details of what's actually happening down at the molecular level, for decades, that's been a total black box, a mystery. And that mystery brings us to a huge problem that scientists have been wrestling with for a very long time. It's the reason the black box has stayed shut for so long. For more than 40 years, if a scientist wanted to get a good look at one of these important proteins in the cell membrane, they had to do something pretty brutal. They had to use harsh detergents to basically rip the protein right out of its natural home. This slide nails the analogy perfectly. The old way, it's like trying to understand a fish by yanking it out of the water and laying it on a table. Sure, you can see the fish, but you learn absolutely nothing about how it swims, how it breathes, or how it interacts with its world. You lose all the vital context. So yeah, you can see why this was a huge problem, right? Those detergents would often damage the protein itself, they'd wash away all the important neighbors like cholesterol, and they'd break apart the very connections scientists were trying to study. It gave them a blurry, incomplete, and honestly, sometimes a totally wrong picture of what was going on. But like in all great science stories, hitting that wall for so long is what led to an absolutely brilliant solution. A real breakthrough that, believe it or not, came in a tiny little bubble. Instead of that violent rip-it-out approach, a team of researchers figured out a much gentler way. They developed a method to get the cell to naturally form these little bubbles, or vesicles, of its own membrane, keeping the protein tucked safely inside. They figured out how to build a mini aquarium for the fish. And this image, which is straight out of the research paper, shows just how elegant this new process is. They're not using harsh chemicals. They're gently encouraging living cells to create these little pockets of their own membrane, with all the important machinery still perfectly in place. Let's walk through it real quick. First, they treat the cells to make them form these bubbles. Second, they use sound waves, kind of like a super gentle blender, to break the big bubbles into smaller, bite-sized ones. Third, they used a sort of molecular magnet to grab only the bubbles with the specific protein they want to see. And boom! It's ready for its close-up, completely intact in its natural home. So what did they see when they finally got to peek inside this perfect little bubble? Well, for the first time ever, they saw things that had been completely invisible before. The level of detail they got is just staggering. We're talking about a resolution of 2.7 angstroms. Now, that number might not mean much to you, but get this. A single human hair is about 500,000 angstroms wide. This is true atomic-level detail. We're talking about seeing the individual atoms that make up the machinery of our cells. And what did this incredible new vision reveal? Well, first, check this out. They saw a literal wall of cholesterol molecules, highlighted right there, plastered all over the outside of the protein. This is a direct, intimate relationship that no one could ever see with the old methods. And then they saw this. A key lipid molecule in the membrane wasn't just hanging out nearby. No, it was actually reaching in and directly touching a critical part of the protein's internal machinery. This hints at a whole new way these proteins are controlled and regulated. And maybe the biggest whoa moment of all was this discovery, a completely unknown docking station for an ion, probably sodium. Think about that. It's like finding a secret on-off switch on a machine you've been studying for decades, a switch you never even knew was there. So what this all adds up to is just a whole new world. By finally seeing the protein in its natural habitat, they didn't just see the protein better. They saw its entire neighborhood, the cholesterol, the other lipids, the ions, and how they all work together in a beautiful, complex dance. And this isn't just one cool discovery. This technique blows the doors wide open for a whole new era of science, a new molecular frontier.
Remember that black box we talked about at the beginning? I mean, just look at this quote. It's from a paper published just a couple years ago. Top researchers in the field were still saying, flat out, that the exact mechanism for how this all works remains unclear.
That was the reality, until now. This breakthrough is the key that finally unlocks that box. So why is this such a big deal? Well, it's a total game changer. It means we can finally study super sensitive proteins that just fell apart with the old methods. It preserves the entire local environment, so we can see how these machines really work inside our bodies. And that, of course, opens up a universe of new targets for developing smarter drugs to fight disease. For 40 years, we were studying the fish out of water. Now we finally have the aquarium. We can see life and disease at a level of detail that was once pure science fiction. And it just leaves you wondering with this one thrilling question. What other secrets are our cells just waiting for us to discover?